Well, good afternoon or welcome everybody. This is Peter Bauer from CENTOGENE. I'm the Chief Scientific Officer and thank you for joining today to go through a webinar where we will talk on cancer immunotherapy through neoepitope vaccination. I think that's a very interesting and thrilling topic, and very interested to guide you through background information and an approach we have made most recently with a project we call PICoP Global, which is an international collaboration assessing neoepitopes for patient-specific vaccination in colon and pancreas cancer. So once again, welcome everybody, and let's just jump into the topic. I think you're aware that cancer is a very relevant disease with high incidences, millions of new cases worldwide on colon carcinoma and a couple of 100,000 for pancreas carcinoma. While the diagnosis is different, severity of the disease, at least in later stages, is highly demanding, and we lack efficient therapies and thereby, of course, have a high burden on society and healthcare systems. You can see that early stages of colon cancer are pretty well manageable, and more than 50% survive 5 years of disease, while in contrast, pancreatic cancer is a very severe disease with an impaired prognosis, and most of the patients diagnosed with this cancer die. What we noticed in the last years is that the incidence is rising from lower levels to the westernized high levels in Asia and South America, which tells us something about contributing factors that probably have to do with Western style of living, and therefore those regions, geographic regions, they will suffer additional disease burden in the next years. Now, when trying to fight cancer, we rely on concepts that have been developed almost 100 years ago, and Paul Ehrlich, one of the godfathers of immunology in Germany, already conceived that tumor cells trigger response of the immune system, and thereby cancer cells might be eliminated through that system in early animal models he had at his hands. Later, of course, this was refined, and meanwhile we learned a lot about as well studying patients with rare diseases with specific immune deficiencies, that they have a significantly increased risk developing different types of cancer, and thereby we have another proof that the immune system is crucial to keep us healthy, and in case we develop cancer, to find a route to reinitiate a proper immune response against these changed cells. If we look into that in more detail, and this is a cartoon where you can see early stages of tumor development and late stages, I think it's kind of a balance where we can see that usually cells that do not behave well and start dividing in an uncontrolled manner usually are recognized by our immune systems, especially CD8 and CD4 positive T cells, and then attacked and destroyed. With developing tumors, sometimes they start to escape this immune reaction and induce an immune tolerance, which gives them a microenvironment where cells can divide and the tumor can be propagated. And so this balance between pro-immunogenic and anti-immunogenic regulatory effects is very important to understand and to manipulate in those immune therapeutic approaches. 3 years back, I think one of the major breakthroughs in tumor therapy, which was the checkpoint blockade inhibition, was proposed at the ASCO and labeled as one of the breakthrough discoveries of that year, maybe even of the decade, Because what we learned, and which is depicted here on the right side in the cartoon, is that advanced tumor cells usually downregulate T cells through the PD-L1, PD-1 receptor complexes, and thereby stop recognition of the tumor-specific antigens by T cell receptor-mediated T cell activation and downstream processes that allow killing of tumor cells. Now, and that's the breakthrough, with small molecules that either block the receptor complex on both sides of either tumor cell or T cell, allowed to restart this immune response and thereby reactivate the patient's innate immune system to fight the tumor cells, which is depicted on the right side. And with this knowledge, of course, a huge field of tumor immunotherapy has been stimulated and developed in the last years. One step back, we can have a model on tumor development as a result of DNA damage. So usually, as shown here on the right upper part, a tumor cell will acquire a couple of specific DNA changes that change the biology of the cell and especially stop cell cycle control mechanisms and promote uncontrolled cell division. Moreover, as well, these changes in the proteins resulting from the DNA changes, of course, have an effect on the surface markers of tumor cells. And there's a very tight quality control system handed over by the MHC receptors that makes these protein changes tangible for immune cells. And so usually these altered proteins presented transiently on the cell surface via MHC receptors are targets for circulating CD8 positive T cells, which then recognize these new proteins, peptides, so-called neoantigens, and initiate toxic, cytotoxic cell death of the tumor cells. This is the basic principle how vaccination against tumor neoepitopes can exert a functional effect, and I will show you in the next slides which kind of proof we have gathered in the last years that this type of therapy is specific and efficient. More generally now, the innovative therapeutic approach would mean that within the tumor cell, in this example, glioblastoma, we have a variety of new tumor-specific mutations giving rise to epitopes that can be seen at the surface of the tumor cells. It's not only one molecule that is altered, it's a couple dozens, hundreds of altered proteins, and so you can expect that tumors during their evolution will have an ongoing change in their surface characteristics. Now, if these changes are identified through molecular mechanisms, molecular diagnostics like DNA sequencing or peptide characterization through mass spectrometry, you can now mimic these surface characteristics through a peptide in vitro production of peptides, which then are administered to the patients like in a virus flu vaccination shot. And then you can recruit the immune system towards these epitopes because now there is sufficient exposure to the immune system, which will recruit formerly exhausted T cells or allow for new mechanisms in fighting these cells. What is clear from this cartoon is that every patient will develop private mutations since they all stem from stochastic DNA events. And so the cocktail, the peptides you need to prepare for this vaccination will be patient individualized. This is very innovative, but as well was very challenging. Nonetheless, the value of the new therapeutic paradigms has been recognized already last year by awarding James Allison and Tashuko Honju for their discoveries in cancer therapy and negative immune regulation, which is in essence the innovation of the checkpoint inhibitors. Now let's look at the published evidence, what has already been shown in literature to be therapeutically efficient in patients with cancer. And one of the first studies that showed that the vaccination against melanoma raises an immune response has been published by Ott et al. In Nature 2017, where they prepared specific peptide vaccination for tumor neoantigens and could show in an immune response that many of these peptides raised a specific response of the cellular immune system against these neoepitopes, either CD4 positive or CD8 positive cells, while the wild type peptide was not raising immune response. So this is a very promising proof of concept study where even more than 50% of the vaccinated melanoma patients had no recurrence within the next 2 years, so giving not only a functional effect but as well as some real measurable benefit for the patients. This approach has been further exploited applying, for example, RNA vaccine. The investigators didn't apply peptides, but RNA molecules that coded for peptides, once again in melanoma, and in the study of Sahin and colleagues from Mainz in Germany, the whole process has been demonstrated first in human as well in 2017, once again in melanoma. And there you can already recognize part of the workflows we anticipated for PICoP Global as well. We receive tumor samples, sequence, and then derive a prediction of potential neoepitopes, which then are put into this personalized drugs, in this case with an RNA vaccine. And these drugs are applied over a couple of weeks in the patient to raise immune response. As said there as well, quite successful. In Tübingen, in my former academic group, we have worked on cholangiocarcinoma and published first patient in 2016 with metastatic cholangiocarcinoma, where the sequencing of the tumor and then the vaccination with peptides was tested against a couple of neoepitopes. And in this very severe and advanced tumor disease, at least we could have stable disease over 3 years in this female patient with a positive immune response against the peptides administered, and as said, clinically a healthy patient without detectable tumors over 3 years. That's promising. And the last study we've seen in malignant glioblastoma, published late last year. A similar approach was taken by colleagues in Heidelberg with collaborators in 2 other clinical centers, where after tumor sequencing, the mutations have been once again identified and predicted for neoepitopes in glioblastoma, and a patient individual vaccination cocktail has been defined, produced, and applied in patients in a subgroup of this clinical study. Overall, successfully on a clinical outcome, so the progression-free survival was more than 1 year in this, once again, very severe aggressive tumor disease. And the analysis as well showed that longer survival was trended to be present in patients with a strong immune response. In this study, a detailed immune monitoring was done and showed once again that the vaccination really activated the cellular immune response, especially CD4 and CD8 positive cells, and thereby complemented a second approach in this study where out-of-the-shelf non-neoepitope-based vaccination was done, which in most of the cases only raised a CD4-dependent immune reaction. So clearly, there's synergy with other state-of-the-art or experimental therapies, and this can be exploited usually with a very low fraction of adverse effects because these peptides are sometimes causing anaphylaxis, but other symptoms are rather like very weak symptoms of a flu, which are usually tolerated by the patients. Now, how did we design to implement such an approach in a global proof of concept study? So the outline there was that we wanted to establish the data protection, which is sequencing of DNA and analysis of tumor material with mass spectrometry to come up with a very robust and fast prediction of neoepitopes. That's the so-called pilot study conducted by CENTOGENE with clinical partners. And in the second step, on the right side, we will partner with treatment opportunities, which we define with Miltenyi to come up with a cell-based treatment model based on the great expertise as well Miltenyi exerts with analyzing immune response and applying cell-based therapeutics. Now, for PICoP Global, it's mainly focused on the first part of the study, the pilot study, where we established the technology and come up with a good prediction of neoepitopes in patients. And this is something I would like to share with you now in more detail. The challenges there are, of course, that the precise identification of neoantigens is still depending on a solid and high-quality data production, especially mass spectrometry is always limited because tumor samples are small. Moreover, you need RNA validation of DNA variants, which is often limited as well because RNA is decayed in clinical samples. And finally, you need a feedback loop to show that the selected predicted neoepitopes really are able to trigger immune response. And so the combination with functional assays, patient-derived functional assays, is very important and part of this proof of concept study. Further down the road, of course, the definition how to synthesize peptides under which regulatory requirements is a major challenge. And in the end, of course, when it comes to clinical studies and to clinical application of this technology, time is extremely critical since these patients are critically ill, and we don't expect that we will have more than 6 to 8 weeks for the whole workflow from receiving the sample to sending out the patient individualized drug or therapy. As said, this is only possible with strong partnerships, and therefore, well, the collaboration and the integration of clinical partners is so ultra critical. What we started in PICoP Global is sampling patients with colon and pancreas cancer after informed consent, where we receive a tumor sample, adjacent normal tissue and blood samples, and a whole battery of omics is then produced by CENTOGENE for these tumor samples and normal samples, which is exome sequencing, RNA sequencing, proteomics, a couple of special analysis, including copy number analysis, sometimes bisulfite sequencing, tumor cell cultivation, and a large fraction as well, biomarker and neoepitope analysis through direct proteomics. linked to the data we have from the tumor samples and the normal samples. And then a third arm is the monitoring of patients throughout disease by analyzing liquid biopsies containing cell-free DNA and as well doing immune monitoring and, where possible, dendritic cell cultivation for more functional assays. This can be broken down in different stages of this workflow, and so for the samples, of course, it's critical that the fresh sample is then assessed, collected, and shipped within 24 hours after surgery to the laboratory so that we can harvest still living, viable tumor cells with representation of neoepitopes and, of course, biomarkers as well that help us to understand patient-specific differences and, of course, as well similarities in all patients. On top, there's a couple of reference blood samples that are needed for the immune profiling and for cell-free DNA monitoring, and as well, of course, within the study, there is the collection of these tumor samples. A part of them is frozen or cooled, and other parts are taken in a specific tissue storage solution, which allows tumor cell enrichment in our laboratory. The DNA workflow and RNA workflow includes cell enrichment step, which is an innovation here in the workflow where we can bring up tumor content of samples from 30 to up to 70, 80%. This tumor cell enrichment is so important to have a higher quality of variant lists and more specific findings in the tumor tissue, and thereby, of course, in the end, a more precise, more specific prediction of neoepitopes. Everything is dealt with next-generation sequencing, which is a highly standardized and efficient process in our laboratory, and these technically, the whole data set is produced within a few days, which really meets as well the expectation in terms of turnaround time. As for the protein, processes are still less standardized, and we are putting significant research efforts in optimization of protocols. There, it's about direct demonstration of neoepitopes, which deals with cell disruption first, digestion of tissues, and then a purification for the peptides we would like to see, and then ultra-high sensitive mass spectrometry technologies to have a very comprehensive picture of small peptides, including the neoepitopes we are looking into. The whole picture in terms of analysis will result in variant lists and as well medical reports for the referring physicians where they can act on standard druggable targets with small molecules, for example, tyrosine kinase inhibitors if possible. The analysis of RNA, protein, and the combined prediction gives then the final output of this study samples, which is the neoantigen prediction list. And this is in a later stage when a clinical study is added, but not within this first phase of our analysis, where then as well treatment and vaccination will be offered. Now, PICoP Global already started, so we have a strong support and partnership with physicians from Pakistan. Professor Chima, the president of the Surgical Oncology Society in Pakistan, joined forces, and now we are collaborating with the University of Lahore Cancer Center, the General Hospital in Lahore, with the Sheikh Zayed Hospital as well in Lahore, and Jinnah Hospital, and services in Faisalabad and the Rashid Latif Medical College. That's a strong group of physicians that trained on the protocol and provide samples within the research project. And of course, we gain a lot of insights into logistics and how qualities and workflows have to be established if you include partners not around the corner. That's something I can share with you because we have just put together a couple of first data from samples, these Pakistani partners. One is whether the cells are still fresh and viable enough to apply the tumor cell enrichment. And you can see that the samples we received from Pakistan have very good uptake of total cell counts in this enrichment. And we can as well see that we improved the number of tumor cells through this technology positively in the Pakistani samples, comparable to other samples we have collected with regional partners. So that's very promising because the logistics are so important in this high-end data production multi-omics projects. And what we could see after sequencing those samples, we have 4 patients with different diagnoses, but we could, for example, show that a patient, one with colon cancer, had more than 3-fold increase of tumor cells through the protocol. He is an HPNCC patient with Lynch syndrome, and so the number of somatic variants was very high, while in patient number 2 with pancreatic cancer, we've seen that enrichment and variant counts are low, but there even enrichment from 2 to 7% already gives significantly better data, since the dilution factor is just close to noise if you start with 2%, while 10% tumor is already handleable in our technologies. We checked as well the RNA quality, and we're not surprised to see that, of course, the bulk tumor cells after shipment had a very degraded RNA, as did as well enriched tumors from those shipped cells, because they even took longer to be prepared. However, the cells still were viable, and after cultivation, shown there on the right side, we could show that the enriched and cultured tumor cells recovered with very good RNA quality, and this will be the source for the RNA-seq validation in the project. Finally, as well, the cell-free DNA can be received in a decent quality. Here, the target size of DNA in the green box is met in many samples. Not all of these samples are really high tumor burden patients, and therefore already the starting material of cell-free DNA is very low in some cases, but there's reasonable amounts in the cohort. So to summarize on the PICoP global project, I think there's no doubt that the motivation to explore patients for individualized therapies is a paradigmatic shift in cancer precision medicine, and promises to offer therapies with less side effects and more efficacy in the future. So we are very, very motivated to contribute in that field. We need these global pilot studies like PICoP Global just to start demonstrating that we can organize robust sample workflows, communication, consenting in different countries, and still offer access to as well new types of therapy, not only in Europe and in North America, but all over the world. Moreover, of course, the diversity of the patients we see, comparing German patients with Asian patients, will help us as well to understand differences and predict the best therapeutic options, depending as well on the genetic background and, of course, the organizational background of these patients and the institutions, physicians we collaborate with. And finally, what we learned is that the earlier inclusion of developing countries in such research initiatives, of course, help us a lot in the communication and educational aspects in genomic medicine, and thereby make us much more efficient in translating that the later stage back into helping the patients with their diseases. Having said that, I think I'm grateful for your attention and I would ask you to put some questions using the Q&A tool of the Zoom software. On top, I have to thank the European Union for funding of this project and would invite you to fill out the questionnaire you will receive and give us feedback on this webinar and how you felt comfortable and whether your interest has been covered. At that stage, thank you, and we have time for a couple of questions. There's one question dealing with this enrichment step of tumor cells, which of course is giving us quality in terms of less noise and more crisp profile of somatic variants. Question is whether this can as well introduce variants that are not found in the tumor. Yes, there's a risk that this might happen, and therefore we just use this source as a primary prediction source, but will always validate in the bulk tumor as well. So variants we only see in the enriched tumor cells don't get the highest score for selection. There's another question asking whether this always has to rely on fresh samples, and the very short answer is yes, since we can only enrich living cells. So we have roughly 24 to 48 hours before the sample needs to be handled in a laboratory, otherwise we lose too much of the precious tumor material. On the other side, of course, we've seen that even with FFPE samples, we can do a lot of validation for the tumor-specific variants, and sometimes even the FFPE samples help us to understand better tumor heterogeneity or consistent markers of the patient in different tumor locations. So there is a question whether this is kind of a collaboration exclusively with Pakistan. At the moment, Pakistan surgeons are the first partners who joined in and gave us as well experience to train the workflows, but we have now prepared logistics and the criteria how to onboard other interested physicians, specifically, of course, surgeons, because we need tumor material. And so there's not a limitation where to offer this collaboration. However, since it takes quite some burden and investment to open up a clinical site, should be reasonable in terms of expected patients to be recruited. Otherwise, probably it's too much work. And there is another question whether in the MENA region we have already partners. We do not have that at the moment, but if there is interest, just get in touch with us and we will evaluate whether we can find a solution or collaboration in that project. If there is no further questions, I once again thank you all for your participation. Don't be surprised if you receive a questionnaire as said, and so happy to stay in touch with you and use the opportunity. If there's any follow-up interest, just drop me an email and I'm very motivated to join into a communication with you directly. Thank you this afternoon and have a good day. Bye-bye.